Just to check, you guys can see my slides?
Yeah, looks great.
Okay. Okay, so hello everyone. So thank you for coming to my talk. I'll be giving a talk today about my journey at the National Center for Functional Glycomics under the mentorship of Dr. Richard Cummings. I started off with parallel synthesis of glycopeptides, and then I've moved on to generating glycan arrays and also doing some work in Glycan informatics. And so I'll be talking a little bit about all these three aspects which we have developed over the past few years. So the first part I'll cover is glycopeptides. That was the main reason I joined the National Center for Functional Glycomics, was to generate libraries of glycopeptides. So to give you a background, glycopeptides can have numerous applications. They can be used for epitope mapping. They can be used for screening of various proteins for binding. They can also be used as mass spectrometry standards. And they can also be used for drug development as well as diagnostics. Classically, synthesis of glycopeptides was performed on resin using solid phase peptide synthesis. If you are familiar with solid phase peptide synthesis, there's not much difference when you're dealing with glycoamino acids in the solid phase peptide synthesis on resin. So basically, you have a resin with an amino acid, and this amino acid usually has a FMOC group at the amine or a protecting group, which can be released. So the resin amine is deprotected, and then the amino acid which is to be added is activated and is reacted with the resin. And this cycle is repeated a number of times to elongate the peptide chain. And finally, the side chains are deprotected, and the resin is the peptide is cleaved from the resin. Normally, the glycan part of the glycoamino acids have acetyl groups on it to protect it from side reactions, and therefore those acetyl groups are then deprotected following resin cleavage. And usually this resin-based synthesis is done in milligram scale. So you do not have a way, or it's not easy to get microgram quantities, and I'll discuss why in the next few slides. So there are several challenges when you're dealing with glycopeptide synthesis. The first thing is the glycoamino acids. Firstly, they are very costly. So this is just some survey which we conducted back in 2019 of different vendors and how much their prices were per gram in terms of US dollars. And for regular serine and threonine analogs, which are used for solid phase peptide synthesis, you could get $2.8 per gram of serine or threonine. But as soon as you start adding any kind of sugars to it, the price really dramatically increases a lot. And because the availability of these the cost of these glycoamino acids is very high, you have to be vigilant about how you use these glycoamino acids to generate libraries of peptides. The next issue is also availability. In this plot, I'm just showing you what is the chemical space which is unexplored in terms of the number of glycan structures which are known. And this was again back in 2019 when we just checked. Glycan has over 14,000 N and O-link glycan structures. Obviously, there is a lot of overlap between them, and there might be cases where there are unknown linkages and those kind of things. But just to give you an idea, there are only 128 glycoamino acids available from Sigma Rich, Sussex, and Glytec. These are the major vendors for glycoamino acids commercially. And so that's another issue, that whatever reagents you do make chemically in your labs, or which you obtain from others, they are a very rare resource, and so you want to use it in a very careful manner. and at the same time get the maximum information out of it. The last issue is lability. Some glycans are susceptible to be labile in the conditions for glycopeptide synthesis, which makes it more challenging. The issues with resin-based solid phase peptide synthesis is that you are unable to use small amounts or volumes of glycoamino acids efficiently. So normally these resin-based peptide synthesis methods are built for larger scale and not for small scale parallel synthesis. There are some instruments out there which you can use to make things in parallel, but usually it is done at a larger scale and in single batches. So I wanted to give you a highlight. When I joined the lab, this was an issue, right? We wanted to generate libraries of glycopeptides, but we had very limited reagents available. And therefore, we wanted to maximize the number of peptides which we could make using this. So spot synthesis is an old technique. It was actually reported originally in 1992. And it's very fascinating how it works. You basically use a filter paper, and it is activated by simply coating the filter paper with an FMOC derived amino acid. Usually that's alanine, and that's by a ester bond. And once you have that amino acid on the surface, you can basically build uh, peptides as spots by just pipetting activated amino acids in different spots and then going through the cycles of deprotection and repeating spotting again. So each spot on these membranes, in essence, elongates to form its own unique peptide structure. And this can finally be deprotected, and the side chains can also be cleaved. And there is actually methods for how to release these from the surface as well. Because if you think about it, it's just an ester bond, so you can quite easily release these from the surface. But this classic method of spot synthesis was not very easy. We tried this obviously, but it was not conducive to glycoamino acids because there were several issues with it. Firstly, the original methods described was kept in open reaction vessels, and usually glycoamino acids take a longer time to react at room temperature, and this makes it susceptible to moisture attraction, which causes side reactions and incomplete reactions and inefficiencies. There was also a lack of monitoring methodology. So to ensure completion, usually when you're doing this spot synthesis, there was a staining method which is involved, which tells you if you did get some uh, uh, um, reaction, but it does not tell you to what extent the reaction is complete. It's a very qualitative test. And the last issue was that the ammonia release, when you try to release these peptides from the surface, it could cause beta elimination and other issues with glycopeptides, especially Olin glycopeptides, right? So we developed a protocol for glycospot synthesis, which is more robust and can be used for a number of different types of post-translationally modified glycoamino acids also. So the first thing we did was, it was very simple. We just bought a glass food container from, Target, for example, and we get these ones with seals which you can just snap and lock on. And we did the entire chemistry in that, in terms of spotting and all. And at the same time, we had a funnel on top blowing nitrogen over this so that it creates an inert environment. And then as soon as we close it, we can turn off this nitrogen and leave the reaction to go on in this vessel. We also used NMP solvent. So usually the classic methods of spot synthesis discuss DMF as the solvent, but we used NMP for these reactions because that has a high boiling point and it also has lesser side reactions with moisture. Um, so we repeated this and we would generate glycopeptides on the surface. But in order to test whether these glycopeptides were being generated efficiently, even before we moved on to the next cycle after putting on the glycoamino acid, we could do something called biopsy testing. So in this, we basically take a biopsy needle and we actually punch a hole inside this spot, which is 1 millimeter in diameter. And this spots of cellulose filter paper can then be taken in an Eppendorf tube. And then using this Eppendorf tube, you can actually deprotect and release the glycopeptides and then analyze either by MALDI or any MS characterization or even HPLC. And the last um, optimization which we did was the release and deacetylation optimizations, and these were done to minimize beta elimination. So we tested a number of different release conditions, obviously starting with ammonia, but then we went on to several different other chemistries to check which one worked the best, and we reported the one which worked the best in all our experiments, and that was actually sodium bicarbonate in methanol water. So using this method, we produced several libraries. The first library which we started off with was a glucanac or glycosylated peptide library. And this was based on a bait sequence by Michael Boyce's lab. In this sequence, we tried to keep it simple, but at the same time, we underestimated the challenge because it turns out that glucanac beta 1 serine is very susceptible to beta elimination. And this actually gave us a good handle to try out various conditions for release to optimize. And in this sequence, this is just a prototypical sequence, but we changed the amino acids across this sequence to a number of different things to produce 20 different peptides. After we optimized the method with the GP1 library, we went on to the GP2 library, which was 40 glycopeptides. In this library, we tried to really see how robust the technique was once we optimized the method, right? So here we started doing things like trying different linkage types. So obviously the O-linked threonine, but then we had cysteine-linked sugars, as well as asparagine-linked sugars synthesized in this using the same method. We also try to incorporate non-natural amino acids. So we replace these prolines over here with fluoroprolines, and that also worked pretty well. And then lastly, we also checked if we could label the N-terminus with fluorophores. So we linked on fluorescein at these two at the N-terminus of these sequences. And in a new paper which we are about to publish soon, we are producing we produce 61 glycopeptides. These actually are the human PSGL1 sequence as well as the mouse PSGL1 sequence. And we even were able to incorporate tyrosine sulfate into these sequences and produce sulfopeptides using this method. So where do we use these libraries of glycopeptides? Well, the first application which we used it was to probe antibody specificity. So CTD110.6 is a very well-known antibody which recognizes glucanac O-link glycans, specifically glucanac beta 1 serine or threonine. So we tested this. The first panel over here is the GP1 library with all the glucanac containing peptides. And here you can see that this particular peptide, it had an acidic glutamate residue before the serine O glucanac. And that caused a decrease in the binding for some reason. But we also noticed the same thing in our second GP2 array. So in the GP2 array, we had much more variation. These are all GalNAc containing peptides where we don't see any binding. And the yellow and green are the glucanac containing peptides, either cysteine linked for the yellow or the green ones are the O-link containing ones. And here too, we see that an aspartic acid residue after the cysteine or serine O-glucanac does cause some does not cause as much of an issue as compared to the glucanac before it. But the CTD110.6 also binds the cysteine S-glucanac with equal efficiency as well. We also use these arrays to check for non-specific binding. So these microarrays, when we printed it at a higher concentration, we noticed that this CTD110.6 started showing non-specific binding. And when we did the same microarray study in the presence of GalNAc, we saw that this non-specific binding to GalNAc containing compounds dramatically reduced. The second application was to use it to interrogate enzyme specificity. So in this application, we checked for the enzyme specificity of ST6-GalNAc1. ST6-GalNAc1 is an enzyme which can put on the sialic acid at the 6th position, at alpha-2,6 sialic acid on the GalNAc or TN antigen to produce the STN antigen. And what we did was we took the same microarrays, and then we either had no treatment with enzyme, or then we treated it with the enzyme at increasing concentration, and we did lectin staining with VVL. Now this VVL lectin recognizes this GalNAc. So you can see that in the starting, when all these peptides had the GalNAc residue, you can see good binding. But as the reaction goes forward and you put on this sialic acid, the binding reduces for all these peptides. And finally, you can see that if you add more enzyme, there is very little binding, suggesting that all of the peptides are converted to the sialic acid derivative. The difference, obviously, what we noticed was that the serine containing or GalNAc containing peptides, which are shown here, seem to be more resistant or slower in reaction with these enzymes as compared to threonine or GalNAc. And there are previous reports of this phenomenon with ST6-GalNAc1, which shows a structural reason to why this happens. So it was good to see this being validated using our methods as well. We also checked GCNT1, which is the enzyme which puts on the glucanac on the core glycans. And in this case, we were using MALDI and HPLC to monitor the reactions. And here too, these are the threonine containing glycopeptides, the first two ones, and this one is serine containing. And you can see the threonine containing, this is before the reaction and this is after incubation. So you can see the shift and the increase in the mass. Here you can see it as well. You can see a little bit of starting product. But in case of the serine containing, you can see that the reaction is only 50% complete. And then we had another application, which was fluorescence polarization. So because our peptides had fluorescein on them, we could also use it to check binding affinity. And we use this using fluorescence polarization assays. So basically we took our peptide at fixed concentration and titrated it against a protein, in this case VVL, and we were able to get the binding affinity using this method as well. And you can see that the cysteine containing sugars were not as well binding as compared to the threonine or serine containing sugars. So to summarize this glycospot synthesis, when do you want to use glycospot? In my recommendation is that if your starting material is limited, if you're doing if you want a small scale of reaction, and if you have a library which you want to generate, which is greater than 10 peptides, and you do not have the consumables for resin-based peptide synthesis, then definitely consider glycospot synthesis. Resin-based synthesis does have its place. Okay, there's no discounting it. When you have starting materials which are abundant, or you need large-scale synthesis, and you don't need a large library of peptides, and you have existing consumables or machines for this, then definitely you can check out resin-based synthesis. There are equipments available for both. However, if you're generating libraries less than 50 glycopeptides, you can still use glycospot synthesis and generate this library. And the potential uses are a little, you can obviously expand on these uses. So basically you can use the glycospot because it produces an array. You can use it for epitope mapping, specificity testing, standards for MS. But you would use the resin-based methods for if you wanted larger quantities. And the uses for those would be like studying in multiple assays if you wanted to do animal studies or drug development. And my recommendation is that please do check out the paper. It has very detailed method section for glycospot. It is easy to adapt in labs where chemistry is not frequently done as well. And we are happy to help you try it out as well as troubleshoot with you. The second part of the story is microarrays. So in 2000s, I joined the lab in 2017, but in 2018, I started doing my own microarrays and printing. And so I'll share a little bit of my experience with that. So what are Glycan microarrays? They are a fundamental tool for investigating specificity of glycan binding proteins. It involves the preparation of glycan derivatives. So you have your glycan and it is attached with a linker or protein or lipids. And these are printed on a slide using a special equipment, which is a microarray printer. And you can put these, you can print arrays on these slides in different patterns, such that each of these sub-arrays can be used for interrogating separate proteins for binding. So this is a miniaturized ELISA in effect. So imagine this is a surface of one of the sub-arrays, and you can incubate a protein on top of it, but you can also incubate other things like viruses, antibodies, even bacteria on this. And then you can add a detection reagent on top of this, and then you can check this fluorescence signal using a scanner, and then quantify the fluorescence signal to give you a reading of the binding to the glycans which are printed on the surface. There are a number of variations you can do to the glycan arrays. You can have different glycans with different derivatives, various linkage technologies. You can change printing parameters to have more number of spots, less number of spots, bigger spots, depending on what you need. You can change the surface chemistry or the slides which you use, the layouts of the arrays on the slides, the concentrations of and buffers which you use to print, and those kind of optimizations need to be done sometimes to get better printing on the slides. Various samples are used. So like I said, you can use glycan binding proteins, but you could also use viruses or even bacteria on the slides. And then a complementary method of detection must be there in case your protein or virus is not already labeled. And you can have various assay variations in terms of incubation times. You can even pre-treat the slides to completely modify the glycans which are on the surface and interrogate it using glycosidases or glycosyl transferases. And then finally, you can have scan settings which need to be adjusted sometimes, and spot alignments need to be done, and processing of the data. So we've printed a number of different things at our center. Just to give you an idea what I have worked with, we've printed glycoconjugates, obviously linked with chemical linkers, but we've also printed proteins and antibodies directly on the surface. The glycopeptides which I've generated, we've printed those as well. We've also printed bacteria on slides. We've printed various chemicals, glycomimetics, flavonoids, and a number of shotgun fractions such as glycans from the human lung and tuberculosis. And I'll show you some results from some of these. So the first array is the NCFG array, which is a defined glycan array. We have a number of glycans of different types printed on this array. And in this study reported in scientific reports, we reported the binding of IgG and IgM glycans for 105 patient samples, where they were differing in age from 20 years to 60 years. And you can see that there's a large diversity in the antibody profiles of all these patients. And some of these patients have very few anti-glycan antibodies, whereas some of them have really high amounts of anti-glycan antibodies. And the second I would say use case for this was the human lung array. So in this paper by Bird-Liotis in Journal of Virology, we studied influenza on the influenza binding on glycans derived from the human lung, and and in this study we actually found that drift influenza strains, which are those which are more moving towards resistance, they started binding more of polylactic structures and salicylic acid containing polylactic structures compared to the regular strains of influenza viruses. And more recently, we've produced an oligomannose array. This is reported in Science Advances last year. And this array has mainly oligomannose structures, and we use this to check urinary pathogenic E. coli, FIMH protein, which we found to bind this epitope on this by using this array. And we also checked HIV broadly neutralizing antibodies, which are shown here. And you can see that these are much more specific or selective in their binding as compared to this FIMH from the E. coli. And these are the epitopes which we have highlighted as to what it's recognizing on this array. So to summarize the microarrays, we regularly print and analyze diverse structures of glycans on multiple substrate types and with a wide variety of samples. So we have a lot of expertise in this. Our arrays are available for use as a service through the NCFG. You can check out the full list of arrays available on our website as well. And we also do custom prints as a service. So this is something which we started doing recently for a lot of users out there. And the last part I would like to talk to you about is the Glycan informatics. So along with the Glycan array work, which I did, I started doing some programming, and I built some tools. So the tools which we create are made for the web. So that way it is easy to access globally in most modern web browsers. All the work which we did was in collaboration with the Glycan team. The tools have extensive documentation and also video tutorials, which should help you for the most part. But if you do have questions, always feel free to reach out to us. And most of our, some of our tools are open source, and the others which I'm building will eventually become open source as well once I polish the code. So the first tool I would like to show is Glycoglyph. It's a tool which I created to just make it easier to draw and name glycans. It's open source. The source code is available here for anyone who's interested on GitHub. It's also a library, so it can be used in other applications to draw. And the tool page is just glycotoolkit.com/glycoglyph, and you should be able to find it. And it's already being published as well, so you can search for it. The features of Glycoglyph are it allows you to enter the names in either the CFG linear nomenclature, or you can use the graphical user interface to input the structure. In the background, Glycoglyph converts this into a JSON structure, and then it draws the structure where you can customize the drawing method which is used, including the symbol size, the linkage, and the orientation. You can save the structures as SVG files. And you can also get the GlycoCT, which is the more machine readable format, which can then be used in other databases like Glycan, Glycan, and others. It can also calculate the mass. However, this feature is limited to certain monosaccharides. If you enter the name using the CFG linear nomenclature, it can actually check if your naming system is right. So supposing you messed up some branch ordering, it will actually try to fix it and those kind of things. And lastly, it can convert this name into the Glycam notation, allowing you to simply click and go to the Glycam website to see the 3D structure of your glycans as well. So this is just a short 40-second demo of the tool. Here you can see that in the templates, you can choose the template which I chose, the ENCODE, and then you can prepare the monosaccharide by clicking on the monosaccharide button and then choosing the linkages, and you add the linkage to prepare the structure. And then with multi-mode, you can just hold the M key on your keyboard and add the glycans on this structure in a very quick manner. The glycosity is produced over here, and you can copy it using this copy button. You can also click on this get Glycan ID to get the Glycan ID for the structure, and also links to other resources like Glycan, and you can get the Glycam link over here as well. And the tool page again over here for anyone who's interested. The second tool which I built for Glycoinformatics was for the data visualization and analysis of Glycan microarrays. And this enables simultaneous visualization of Glycan structures along with Glycan microarray data. And also it allows you to generate beautiful visualizations and allows you to explore your data and also do the data wrangling inside it as well. And the tool is also on glycotoolkit.com/glad. I won't go into the details of this. There is a very in-depth documentation as well as a YouTube video on this. You can find that video as well as links to the documentation over here. And basically in this video, you can go from any of the CFG data sets in 20 minutes to generating graphs like this with a few data sets from the CFG data. And currently what I'm working on is a data management platform to help the users enter data, the microarray experimental data into the database, and also to connect this database to other databases and eventually feed this data into the Glycan array repository. I'm rushing through the informatics part because I know I'm going over time, and I apologize for that. So just to summarize, this is the journey of creating tools which I've had over the past few years. I started off with glycospot synthesis, but I've also moved on to Glycan microarray printing and assays, and also now informatics. And lastly, I would like to acknowledge the people involved, specifically my PI, Dr. Richard Cummings. He's been an excellent mentor and he's very patient as well, which was very helpful in developing these tools. All the members of the Cummings Lab and the NCFG and the co-authors and collaborators who I've worked with for all these projects. The funding from various sources. And I'm leaving this contact information here. We do have multiple opportunities for postdocs as well as research scientists and technician positions. If anybody is interested, do please feel free to contact me. So I'll stop there. Thank you.